Okay, this is the second part of this study session for test 5. This is just about my last anatomy exam before our final, so I'm really trying to do good, because if I ace this, I'm able to be exempted from the final exam. So, something I've been going since about 5 in the morning. Right now it's about 7:20, so I'm still hanging out in there. We went through a bunch of the notes in the previous video, and I'm gonna try and finish it up, see how far I go, and then we'll jump into endocrine hormones, and then we'll maybe look, if we have time, we'll look at the online study guide, and then we'll take that exam in about 2 hours. Let's go. Bit of a recap, quiz results. So for the quiz, the 5 big types of receptors you need to know were photoreceptors, thermoreceptors, mechanoreceptors, nociceptors, chemoreceptors. Transduction is the converting of a stimulus from the inside or outside the body into the language of neurons. Converting of a sound wave, light, photon, baroreceptor on the form of physics. Our body sends neurosignals, zaps, converted into the language of neurons at the receptor cell. So on the endocrine hormone handout, the first column is empty, regulated by some stimuli that we have to fill in ourselves. Mentality is a cause and effect chain. She wants us to know the immediate effects. Aldosterone, ADH, example. Increased sodium reabsorption. And know the ultimate effect. ADH, increase water reabsorption in order to increase blood pressure. And error was at the parathyroid hormone. Okay, back on that same figure from the previous video of the section of the eye, right there. So the eye has accessory structures: lacrimal gland, lids, conjunctiva. The sclera has dense fibrous connective tissue, bulge in front of the cornea, and outlet in the back of the optic nerve. The sclera has 3 tunics. The fibrous tunic is the outer one. It is made up of the cornea and the sclera, the sclera is a continuous structure where this fibrous connective tissue becomes transparent and bulged at some point in the sphere. Our eyes have different shapes. It can be elongated, bulged, after the accommodation story. The cornea is bounded by conjunctiva in a thin membrane over the surface. The cornea and sclera are made up of a lot of collagen. The conjunctiva contains tiny blood vessels. Bloodshot eyes are when capillary vessels are broken and repair quickly. The bulb is covered with collagen. The purpose of this dense, tough bulb is to protect the delicate things inside, allows for muscles to attach to it. The cornea and sclera are made up of a lot of collagen. vascular tunic is the middle layer. Tunic inside the ball. The vascular tunic is the middle layer, the tunic inside the ball. It's vascularized, also known as uvea tunic inside the bulb. Anterior ciliary. The anterior part of the vascular tunic has a ciliary body, serrated edge, darkened region. Once sliced through, you can see it with little threads at the edge called ciliary processes. That suspend the lens on the ciliary kidney. Oh, I'm falling asleep. Suspend the lens on ciliary body, suspensory ligament, capillary secrete aqueous humor that bathe the front of the eye. Capillary secrete plasma that is filtered and comes out to be aqueous humor that is nourishing and bactericidal. Anterior cavity is divided into two chambers. The anterior chamber is in front of the iris. The posterior chamber of the anterior cavity is in front of the lens. Nuts. The anterior cavity is divided into two chambers. Anterior chamber is in front of the iris, posterior chamber. The anterior cavity is in front of the lens, but posterior to the iris. Aqueous humor exists in front of the lens, which is divided into two pockets, anterior and posterior. The iris is the dividing peninsula, dividing the same liquid. Aqueous humor disc, extensions of the ciliary body that determine how much light hits your lens. Determines how much light. Circular and radiating muscles determine how much light comes in. The iris is responsible for determining how much light comes into your lens. It's controlled by circulating and radiating muscles, controlled by either sympathetic or parasympathetic. Sympathetic is more on the radiating muscles for dilation. Circular muscles more for constriction of the pupil. Parasympathetic division for that. Sympathetic division for the iris is radiating muscles. Contract the eye, opening the pupil. Parasympathetic is circular muscles, closed spaces. Iris is a nice colored part of the eye backed by dark pigments, light absorbing cells. In addition, the ciliary body, the iris, human pupil, there is the lens. Light absorbing cells. In addition, the ciliary body, the iris. In addition to the ciliary body, the iris, the human pupil, there is the lens. The lens is a squash sphere, egg-shaped, attached by suspension ligament. Light comes into the pupil and hits the lens with little threads attached to the ciliary body. Very distinctly dilated pupils means there's nerve damage. The parasympathetic nervous system not operating properly. Next figure is 17-5B. So this figure, we can see the fibrous layer, the three layers of the eye: the fibrous layer, vascular, and the inner layer, the retinal layer. Fibrous layer consists of the cornea and the sclera. Vascular layer consists of the iris, the ciliary body, and the choroid. And the inner layer or retinal layer consists of the neural part and the pigmented part. Okay. The posterior to the eyeball is the choroid layer. Darkened layers of cells, part of the thickened vascular layer. Choroid is inner vascular, where blood supply is brought in and nourishes the retina of the eyes, full of melanocytes that send fingers into the dermal layer. Protect the cell nuclei from sunlight. Choroid layer absorbs stray light. Animals with night vision reflect choroid to the retina. Particle vascular choroid full of melanocytes. Posterior nuclei from sunlight. Choroid layer absorbs stray light. Okay. The third layer is the neural tunic, or the innermost layer, or the innermost tunic. It's about the nervous system, not the vascular system. The retina, or the orange layer, is a very active layer of light-sensitive cells. Transduction takes place in these cells, several layers from the retina, because they are forming converging circuitry. There's lots of receptor cells that are speaking to fewer neurons. There's lots of receptor cells speaking to fewer neurons. Fewer neurons, and eventually combining information, sending them out to the optic nerve, allows you to have extreme sensitivity Lots of rods and cones feed into few ganglion cells and to the optic nerve. Oh, dang! Combining information, sending them out to the optic nerve allows you to have extreme sensitivity. Lots of rods and cones feed to a few ganglion cells to the optic nerve. Next figure. Next figure is on this one, the organization of the retina. So you see the choroid layer, pigmented part of the retina right there. Rods and cones, bipolar cells, endocrine cells, and ganglion cells. Here's a different view of the neural part of the retina. You can see the ganglion cells, then the bipolar cells, then to the rods and cones, then to the epithelium, then to the horizontal cells. But if you want to reach those, the optic disc needs to reach the nerve fibers. Yeah. So this is a picture of a slice of the retina. Sensitive cells, choroid layer in the back. Yeah. There's a choroid layer in the back. There's a retina or a skinny layer. And there's a skinny layer. Optic nerve is a blind spot that has no rods and cones. Rods and cones look different in shape. Falling asleep on camera. This is the second round, so it's kind of tough for me to keep going. Upper layer reaches through pigments embedded into the background of pigmented epithelium. Starts in the back with a skinny pigmented layer. Rod-like receptors and cone-like receptor entities. Next figure. Next figure is right here. So we got some light coming in, hitting the photoreceptors, the bipolar cells, then synapse of the photoreceptors, rods and the cones. Some nuclei, Golgi apparatus, mitochondria, and then we go into some the outer segment, and then we get to the pigment epithelium that absorbs photons that are not absorbed by visual pigments. is also phagocytosis of this shed in a rod. Each disc is an independent entity, and there's an outer segment forming an elongated cylinder where there are structures of rhodopsin molecules there. There's a rhodopsin molecule which has opsin on the inside and retinal also in there. Okay. Let's keep going. Rod-like receptors and cone-like receptor entities embedded into the backbone of the pigmented epithelium ensures that light is reabsorbed back into pigmented layer. Rods are sensitive to shades of gray, black, and white, no color. Much more sensitive to dim light. Picks up light at lower levels. Example, be walking around in moonlight, late dusk. Vision is not good because not enough light is seen. Only a good color vision is in daytime. Information is sent to a layer of neurons inside them called a neuron. Let's start at a junction between bipolar cells. So we're going from bipolar cells to ganglions. Right. Isn't this bipolar cells? Isn't it ganglion cells to bipolar cells to rods and cones to the pigmented epithelium? Bipolar cells pick up information and turn it into an action potential. Bona fide neurons. Cones pick up wavelengths in different colors but require more light. Sensitive rods and cones send info to layers of the neuron. Ganglion cells are in the back of the eyeball. The departure of the optic nerve. We're going from ganglion cells to the back of the eyeball. Departure of the optic nerve is not directly behind the pupil, off center. It has to carry information to your brain. And ensures retina is capable of receiving light. We get a focus image from part of the retina directly behind the pupil. A focusing pupil called the fovea. Retina is capable of receiving light and get focus image from part of the retina. Briefly behind the pupil, a focusing point called the fovea. The macula is a spot in the center containing a middle point called the fovea. The blind spot is where the brain tries to convince us that we don't have a blind spot. It has no rods and cones, no vision at all there. Next is figure. Next figure is on image formation. So light from a point at the top of an object focus on the lower retinal surface. Light from a point at the bottom of the object is focused on the upper retinal surface. Light rays projected from a vertical object show why the image arises upside down. Light rays projected from a horizontal object shows why the image arises with left and right reversal. The image also arises upside down. Yeah. Image formation. Any image you look at will be projected, will have some height, depth, and dimension to your retina as an image upside down. Let's see. Any image you look at will be projected, will have some height, depth, and dimension to your retina as an image upside down, so our brain. And backwards, light is projected to your lens and projected back to your retina in a straight line, so you can see something even upside down. You can only focus on part. You can only focus part of a pole that your eyes focus on. You can't focus on the whole pole. In order to focus on the pole, you have to use muscles to move your eyeball so that it is turning, and directly behind the pupil will be aimed at the bottom of the pole. In order to focus on anything, the eyeball needs to be turned so that it can be projected on the fovea to be focused on. On the retina, it is backwards, and upside down is sent to the brain so it can be seen right side up. Hundreds done. Okay. Next figure is on factors affecting focal distance. Light rays from a source are refracted when they reach the lens of the eye. The rays are then focused onto a single focal point. This is the focal distance between that and the focal point. Okay, closer light source, longer the focal distance. Rounder the lens, the shorter focal distance. What is happening with light? Light is reflecting off itself, bending light and projecting it onto the fovea. The lens bends light and is expressed at the fovea. What's happening when the light is reflecting off itself, bending light and projecting it onto lens bends light expressed on the fovea. Light goes from air to dense fibrous to gelatinous sized. When looking at something from far away, light is reflected off it, so that light has to bend at a certain degree. When looking at something from far away, light is reflected off it, so that it has to bend at a certain degree, bending at an angle. Lens has to do something for the focal points to be at the fovea of the eyeball, to make sure light is bent enough to hit your fovea. And as the lens is rounded, the fovea bends light more. A curved object will bend light, and flat object will send light straight through. This object requires your lens to do different things, causing your lens to curve or flatten, so that your lens will be able to flex and bend light more, curving light less. A curved object will bend light, and a flat object will send light straight through. Next figure is on accommodation for the eye. To form a sharp image at the focal distance must equal the distance between the censored lens and the retina. Close vision ciliary muscles contracted. For close vision ciliary muscles contracted and the lens is rounded. Contracted ciliary muscles and rounded lens. For distant vision, ciliary muscles is relaxed and the lens is flattened. Flattened lens. Relaxed ciliary muscles. Okay. Close vision. Okay, close vision ciliary muscles contract, the ciliary lens is rounded. As ciliary muscles contract, they round the sphere to get light, the focal point, to meet at the fovea, shortening that focal distance. Distant vision, ciliary muscles relax, the lens is flattened. The focal point has to meet at the fovea. Focal point has to meet at the fovea. Tells the lens to bend that so that the ray can meet at the lower fovea at 20/20 vision. Making the lens to bend the light enough so that the rays bend and meet your fovea. All about distance. Focal point meeting at the fovea. The two problems that would occur is that when you're older, your lens is not flexible enough. Lens or ciliary muscle, the lens gets harder, making it harder to round it and meet the focal point. Muscles cannot be rounded enough to meet the focal point or the fovea. The muscles can weaken, the eyeballs cannot be perfect spheres, lens is not curvy enough. These are all part of accommodation. Eyeballs can also not be too long or too short to bring light to the right spot. Using another lens, glasses help rays come in at a straighter angle. An artificial lens for light bending, artificial accommodation with glasses, or contact lens. Those are other alternatives too. Okay. Next figure is on. Back to this figure of the pigmented epithelium and the rods and the cones. Photoreception by receptor cells. Rods are sensitive to black and white shades of gray. Cones are sensitive to different wavelengths of light and different colors of light. Next figure is on photoreception, the states. So sodium entry through gated channels produce a dark current. So we have rhodopsin and opsin's role in all this story. Sodium entry through gated channels produces a dark current plasma membrane in the outer segment of the photoreceptors. Movement of ions to the outer segment. To the inner segment. And out of the cells known as the dark current. Okay, so ions come in. Okay. Different cones. Cones are sensitive to different wavelengths of light. Several different groups of cones always working together. Oh, I was actually looking at cone sensitivity. So this figure right here, down here, this graph compares absorptive characteristics of blue, green, and red cones and the typical rods. Rod has only one wavelength and it's black. The others are colored wavelengths. Cones are sensitive to different wavelengths of light. several different groups of cones always work together. If you have some cones not working properly, then you are colorblind in a certain spectrum. You don't need to know the numbers of visual light spectrum, photoreception, the sense of enzyme-induced steps that take place inside the sensitive cells. Sensitive cells have pigments that absorb different wavelengths of light with a specific absorbance, absorbing certain colors and reflecting the rest. Retinal is a carotenoid association between vitamin A component and pigment. Back to that figure of the rods, cones, and the pigment epithelium. Photopigment absorbs certain colors and reflects the rest. The pigment is called retinal. Retinal. And retinal comes from eating carrots, bruised retinal pigment. Retinal is the vitamin A pigment component. Opsin is the glycoprotein component. Glyco means carbohydrate. Retinal plus opsin produces rhodopsin. Rhodopsin is a glycoprotein. Rhodopsin gets split into two components, retinal and opsin, when hit by light, called bleaching. Bleaching of photopigments. Photopigments get split apart until pigment is not able to receive light. When light hits retina, photopigments split apart. Rhodopsin is constantly regenerated, rebuilds itself quickly, damage and rebuild. Rhodopsin. Rods are more sensitive to levels of light. And take longer to put themselves together. Back to the figure of sodium photoreception 1. The process of photoreception is a process of destruction. In the dark, there is a dark current operating. Dark current is the movement of ions into the outer segment, on the inner segment, and out of the cell. Ion movement. In the dark, there is a dark current operating. Neurons, the sodium gates are open. Process of breaking. In the eye, the current is backwards. Only a little bit of sodium comes into the neuron. In the eye, the neural activity is backwards, taking light in and breaking it apart to individual pieces. It's the difference between rods and cones' ability to regenerate, which is much sensitive. Rods are more sensitive, but take longer to be brought back together. Comes faster. Cones are faster, put opsin together more rapidly. So it's breaking apart and regenerating of many rods and cones. Next figure is of bleaching and regeneration of visual pigments. So we have, after absorbing light, the retinal changes to a more linear shape, activating the opsin molecule. Opsin activation changes sodium permeability, so sodium can enter. The transmitter release the synapse. Changes in the bipolar cell activity. Location of stimulated ganglion cell indicates specific portion of retina stimulated by arriving photons. So neurotransmitter release bipolar cell on the ganglion cell. After absorbing a photon, the rhodopsin molecule begins to break down into retinal and opsin. This is known as bleaching. The retinal is converted to its original shape. This conversion requires energy in the form of ATP. Once the retinal has been converted, it can recombine with opsin, rhodopsin. Molecules not ready to complete this cycle. Regeneration process takes time. After exposure to very bright light, photoreceptors are inactivated while pigment regeneration is underway. So that's bleaching and regeneration of visual pigments. Okay, rods take longer to regenerate. It takes a while before you can see a new image. Cones regenerate fast, regenerate in a few minutes. Visual images relative to light and dark in color. Brighter light means it's more destructive, less time to regenerate. Lower light is dimmer light leads to less destruction, longer time to regenerate. Cones regenerate fast, regenerate in a few minutes. Visual images relative to light and dark in color, it does occur. Rods take longer to regenerate. When sodium gates close in the presence of light, only during receiving until there is light, bipolar cells say to send to the brain of light are bipolar cells inhibited until there is light and sent to the brain. The brain unscrambles it. Brain gives sensitivity and resolution. The eyeball or the retina tells you to contrast light and dark. Cerebral cortex gives you image. The cerebral cortex gives image through the thalamus and integration here. Okay. Moving to bright light from a dark room or the dark to light version, vice versa, takes a while for you to see because rods are destroyed by light. Cones regenerate fast to see color. Oh! Next would be the most latest lecture. We're almost there. Okay. So we had a quiz on the eyes. So the eyes are accommodating. So myopia means you're nearsighted. Hyperopia means your eyes are working better with distant objects. Myopic is a person who's nearsighted. Emmetropic means that you have both eyes working properly. If you're myopic, your focal point is in front of the fovea. Lens is not rounding enough. Myopic for close object. Your lens needs to round more. The lens is rounding, allowing for successful bending of light. Requires lots of bending. So it's on this. More based on this figure of light bending needs to hit the focal point. There's the focal point hitting the fovea. For myopia, your focal point's in the front. Hyperopia, focal point's too far in the back. Okay. For faraway objects, bends focus with not a lot of bending. A faraway object would focus more on the upper part of the lens. When the lens is curved and rounded, it causes your waves to bend more. When it's flat, less bending of light for close objects. Angled light common into two lens. The eye has two choices: to change the shape of its bulb or change the shape of ciliary muscles to meet the fovea at its focal point. The flatter the lens, the more bending. The more focal point is pushed back into the fovea, the shape of the lens and length of the eyeball. Transduction is what happens to turn a signal to an action potential, such as the tilt of a head, gravity, sound waves, light waves. Transduction is electrochemical nervous signal. Sound waves, molecules get compressed, gets into the fluid of endolymph to the cochlea, or to the organs of chordae in the cochlea, then to the basilar, then they move the basilar membrane, then the hair cells bend, causing molecular changes in the cell, forming an action potential. Next, we finally reached the endocrine system, chapter 18. Finally, that took so long. So the first figure is of organs and tissues of the endocrine system. So we have the hypothalamus producing ADH and oxytocin and regulatory hormones. Pituitary glands, adrenocorticosteroid, tropic hormone, thyroid stimulating hormone, growth hormone, prolactin, follicle stimulating hormone, luteinizing hormone, and melanocyte stimulating hormone, all from the pituitary gland. Posterior lobe produces oxytocin and ADH just like the hypothalamus. The thyroid gland produces thyroxine, T4, tриодонine, T3, and calcitonin. That's the thyroid. Adrenal gland produces at the medulla epinephrine and norepinephrine. At the cortex, it produces cortisol, corticosterone, aldosterone, and androgens. Pancreatic islets produce insulin and glucagon. Okay, the pineal gland of the brain produces melatonin, hormone. The parathyroid glands produce parathyroid hormone. Some organs with secondary function include the heart, producing AMP and BMP, the thymus, producing thymosins, digestive tract secretes numerous hormones involved in coordination system functions, glucose, metabolism, appetite, kidneys, erythropoietin, creatine, calcitriol, gonads, testes, androgens, especially testosterone, inhibin, ovaries, and estrogen, progesterone, and inhibin. Okay, the endocrine system. You need to know all those ones in that figure. Next figure is 15-2. Right next to it, structural classifications of hormones. Hormones of the body can be divided into three groups on the basis of their chemical structure. So, first one, amino acid derivatives, small molecule structure related to amino acids. Amino acids are the building blocks of proteins. There is a tyrosine. These include thyroxine, which is produced in the thyroid gland, epinephrine, produced in the adrenal medulla. Sources of tyrosine include meat, dairy, and fish. Derivatives of tryptophan, melatonin, which is produced in the pineal gland. These are all amino acid derivatives right here. Thyroid hormones, catecholamines. Peptide hormones are chains of amino acids, so chains of these guys. Most peptide hormones are synthesized as prohormones in active molecules that convert to active hormones before or after they are secreted. They include glycoproteins, so like the opsin guy that form the rhodopsin, glycoproteins, proteins that are more than 200 amino acids long. These glycoproteins include thyroid-stimulating hormone, luteinizing hormone, and follicle-stimulating hormone. And the anterior lobe of the pituitary gland. They come from there. Short polypeptides and small proteins. This group of peptide hormones is large and diverse. It includes hormones that range from short-chain polypeptides such as ADH and oxytocin, each 9 amino acids long, to small proteins such as insulin, growth hormone, and prolactin, secreted by the hypothalamus, heart, thymus, digestive tract, pancreas. Lipid derivatives, 2 classes: eicosanoids, eicosanoids, and steroidic hormones derived from cholesterol. Eicosanoids are important paracrine factors that coordinate cellular activities and affect enzymatic processes such as blood clotting and extracellular fluids. Some eicosanoids include leukotrienes and prostaglandins for coordinating cellular activities in some tissue. Prostaglandins can be converted to thromboxanes and prostacyclins. Steroid hormones are released by reproductive organs. Example would be estrogen and progesterone, ovaries and female by the cortex of the adrenal gland. Corticosteroids and by the kidneys, cholesterol, because circulating steroid hormones and specific transport proteins in the plasma remain in circulation longer than secreted peptide hormones. Okay, that figure. Going into the notes, we get to know all 3 of those color tags. The endocrine system is the chemical communication system for system-body controls, as opposed to the neural system, neural and hormonal systems of the body. Commanding and controlling everything going on in the body systems, signal the flow of ions across the membrane. Commanding and controlling everything going on in body systems, signal flow of ions across membranes. A molecule of a substance is dropped in the into the bloodstream. And my sweet mate is playing the rap music. A molecule of a substance is dropped into the bloodstream. Okay, I'm awake. Let's go. Endocrine system causes and effects of the endocrine system. In contrast, there's a difference in the structure of the endocrine system and the nervous system. The nervous system works on neurons and cells which connect to the endocrine system. Has a chemical story, deals with chemicals dumped into the bloodstream. The endocrine system has a chemical story. It deals with chemicals dumped into the bloodstream. How does any specificity come out of the membranes? Like specificity of the neurotransmitter, target organs or cell has a specifically sized receptor site. Specificity at target end where not received by every organ. It's picked up only where cells or organ has a specific receptor or molecule that fits the molecular shape of the molecules of that hormone. Some similarities and differences between the nervous system and the endocrine system. The nervous system is hardwired circuitry. The endocrine system is not. The body is producing hormones. Some are stored. No circulatory system, just for hormones, and broadcasted uniquely. So no circulatory system, just for hormones. So the nervous system works on neurons and cells which connect. The endocrine system is more of a chemical story. It deals with chemicals dumped into the bloodstream. How does any specificity come out of the max dumping? Like specificity of the neurotransmitter, target organ or cell has specifically sized receptor sites. Specificity at a target end where not received by every organ, picked up only where cell or organ has specific receptors or molecules that fit the molecular shape of the hormone. Some similarities and differences between the nervous system and endocrine system. The nervous system is hardwired, endocrine system is not. Endocrine system is producing hormones, some are stored. No circulatory system, just for hormones and broadcasted uniquely. There's no circulatory system. The nervous system, it works quickly. Endocrine system is not working very fast. Relies on the beat of the heart to ensure hormones are picked up by target organs or cells. Emphasizes more on duration of a signal. So the nervous system activity ends fast. Nerve signals speed up. Endocrine system has longer duration of effect. Hormones get dumped into the blood and takes a while for its effect to diminish. The endocrine system can be compared to the exocrine system, where a gland is produced by a substance put into the ducts, leading into bigger tablets of output. There's little tubes leading to bigger tubes. An example would be the surface of the stomach, the skin, the eyeballs, and the sweat gland. The endocrine gland doesn't have specificity as they secrete there. Specificity comes with the specificity of the receptor. Where a gland is produced by cells put into the ducts. Many neurotransmitters can behave as hormones. Many neurotransmitters can behave as hormones. Some constituents include amino acids, peptides, and proteins. Yeah, many neurotransmitters can behave as hormones. Amino acids, peptides, and proteins. Lipid story. For the lipid story, template for hormones is based on a template ring structure. Template is based on a ring structure. Example would be cholesterol, not amino acid rings. Bonded rings. Unique structure works differently when reaches the target. An example would be amino acid hormones, epinephrine. Also a neurotransmitter used by the sympathetic nervous system. Also secreted from the medulla on the adrenal gland. Dopamine, thyroxine. Some digestive hormones include peptide hormones. Peptide hormones include gastrin, somatostatin, CCK, secretin. Hormones from the pituitary gland or tropic hormones include ADH, some steroid hormones secreted from the adrenal gland. Steroid hormones are lipid derivatives. Some steroid hormones include from the adrenal cortex has 3 layers, outer, inner, and lower. Sterum is the key to being a steroid lipid hormone. Some examples include cortisol and cortisone. Inner cortex secretes testosterone. When the hormone reaches its target organ the general way, calcitriol is a steroid hormone. How the hormone works? In the general way, hormones are received at the surface of a target cell and are brought in the cell using various molecular activity on some activity in the cell. Okay, next part. They're invited into the cell by molecular mechanisms going into the nucleus and work directly onto on the processes in the nucleus, turning on and off genes. Genetic affecting replication and transcription. Genetic processes that happen in the nucleus. Subnuclear happening in the nucleus. General roots working in membrane, working on cell processes in the mitochondria. Oh, dang. Oh. Working in the nucleus and the mitochondria and ribosomes. Ultimately, endocrine hormones activating enzymes and cause secretion of enzymes. How do these two roots? Endocrine functions might activate enzymes or cause secretion of enzymes. Peptide or protein-based hormones. Pancreatic hormones cause secretion in the duodenum. Examples include gastrin secretion causes secretion of enzymes. These include pepsin, permeability, changes from permeability of membranes. Examples would be the DCT, ADH, aldosterone enzymes that change permeability, collecting duct, and aquaporins. Aldosterone changes membrane permeability. Initiate protein synthesis. You start production of protein synthesis, start production of enzymes, hormones, cell structure, plasma proteins, stimulate secretion of substances. Example would be gastrin and pepsinogen. One hormone can do one or more of these things. Lipid-based hormones can get involved in transcription and translation, really basic signals to turn genetic processes, replicate, transcribe, and translate. Let's figure. This figure is the feedback control of endocrine secretions. So right here, we see typical pattern of regulation when multiple endocrine organs are involved. Hypothalamus, releasing hormone, negative feedback. From the pituitary gland, we get anterior lobe producing a hormone to the endocrine organ to a hormone. That second hormone produces negative feedback to the releasing hormone to stop releasing itself, and finally it stimulates the target cell. The hypothalamus produces release hormone. Stimulate hormone production by upper glands. Control occurs by negative feedback. Variation of the typical pattern of regulation of endocrine organs by the hypothalamus and anterior pituitary lobe. Example would be prolactin hormone, inhibitory hormone, and prolactin releasing factor. Prolactin releasing factor activates in the anterior lobe release of prolactin, and as that is being produced, there's also an inhibition of prolactin factor happening, and stimulation of prolactin inhibitory hormone also happening. Regulation of prolactin production by the anterior lobe, in this case the hypothalamus, produces both releasing factor and inhibiting hormone. When one is stimulated, the other is inhibited. Next one is gonadal hormone. There's a releasing hormone and inhibitory hormone. And then the gonadal hormone affects the liver, produces somatomedins, which stimulate the growth of skeletal muscles, cartilage, and many other tissue. While these are being stimulated, the gonadal hormone inhibitory hormone is being also stimulated, which inhibits the release of gonadal hormone releasing hormone. The regulation of growth hormone production by the anterior lobe when gonadal hormone or releasing hormone is released is inhibited. Gonadal hormone inhibitory hormone release is stimulated. Releasing hormone, thyroid releasing hormone, hormone 1, thyroid stimulating hormone, endocrine target organ, thyroid gland. Hormone 2 is the thyroid hormones, cortical releasing hormone, adrenocortical trophic hormone. The thyroid organ would be adrenal cortex. Hormone 2 glucocorticoids. Next figure is more on the pathway story. So, hormone feedback. A gland is stimulated and the gland responds by secreting something. Secretion is going to stimulate another gland to do something else. Pathway story. Hormone feedback gland is stimulated and gland responds by secreting something. Secretion is going to stimulate another gland to do something else. In hormone chain, an initial hormone released by the hypothalamus goes to another gland and has that other gland release its own hormone, and that second hormone gets to another gland releasing its own hormone. Speaking to a gland, example would be anterior pituitary releasing hormone to the bloodstream. Speaking to another gland, hormone to the bloodstream to target circuitry. Not as simple as stimulus and an effect. 18-6. Anatomy and orientation of the pituitary gland. So the anatomy and orientation of the pituitary gland is like this. So you see the hypothalamus, there's a third ventricle, there's anterior pituitary, you can see the pars superalis, the pars distalis is the anterior, pars intermediate is in between, posterior pituitary lobe, the neural neural hypophysis, adenohypophysis, pars intermedia, and neural hypophysis. So adenohypophysis secretes suprapituitary hormones. Pars intermedia secretes MSH. Neurohypophysis or posterior pituitary lobe produces ADH and oxytocin. Okay. Hypothalamus is an organ of the nervous system, even more than an organ of the endocrine system. It has a connection to two parts of the pituitary gland. Okay, next figure is this one, the three mechanisms of hypothalamic control over endocrine functions. The first one is the production of ADH and oxytocin. ADH and oxytocin, secretion of regulatory hormones to control activity of the anterior lobe of the pituitary gland. Control of sympathetic output to the adrenal medulla. Yep. Okay. So, because it's controlling sympathetic output to the adrenal medulla, it controls the secretion of epinephrine and norepinephrine. Okay, so the neural part is the hypophyseal stand. It's directly connected to the hypothalamus. Neural hypophysis, ADH story. ADH and oxytocin story. Direct neurons runs out of the hypothalamus and straight to the neural hypophysis. So neurons don't go straight to, and the adenohypophysis goes straight to neural hypophysis from the hypothalamus. So there's two hormones, ADH and oxytocin. Hormones travel down neurons and release at the end. The two hormones are ADH and oxytocin. Neural tissue secreting ADH responds to the stimulus comes to the hypothalamus. What causes the hypothalamus to secrete ADH? Well, number one, lowers blood pressure by baroreceptors. Hypothalamus gets the signal and responds. Number two, renin and angiotensin start to circulate and give the same signal. Also gets info from the bloodstream responding to all three signals in the same way. Needs more fluid in the blood. Sends signals down the neuron into the bloodstream. ADH travels into the blood to the kidneys. At the collecting duct and DCT, which increases aquaporins. Aquaporin activity will allow more fluid not to be excreted as urine but to be reabsorbed into the blood. Increasing fluid in the blood increases pressure. Increasing aquaporins don't change in size, they change in number, allowing more fluid to leave the collecting duct. ADH causes more aquaporins, more fluid leaving kidney tubing heading to renal pelvis, back into reabsorption in the blood, direct stimulus in the bloodstream to a gland. Direct stimulus to a gland, then gland to the bloodstream, then to the target. Senses concentration of sodium ions in cerebrospinal fluid. Also has osmoreceptors, has a type of chemoreceptor that senses osmolarity, senses concentration of sodium ions in the cerebrospinal fluid. Okay. Another hormone secreted by this gland, the hypothalamus, the pituitary gland, these releasers and what each releases. Pituitary gland releases oxytocin also. Leaves neural hypophysis to the bloodstream to receptor cells in a lot of places in the body. An example would be in the uterus. Smooth muscle walls respond by contracting, and for mammary glands respond to oxytocin by ejecting milk. Milk can't be pushed out until there is suckling of the infant. The uterus does not start contracting. It has to do with stretch. When the stretch of the stimulus is right, oxytocin starts seriously contracting. Okay, another more complicated than the ADH story. You cannot spurt milk unless you have the stimulus of a suckling infant. The uterus does not just start contracting. It has to do with stretch and hormone levels. Oxytocin is called the bonding hormone. Oxytocin causes surface contraction every month and menstrual lining. Bonding hormone has to do with identification process, has to do with odor, the hypothalamus. The bonding story exists in organisms other than humans. At high levels in children, mothers, infants, fetuses, males, and females during sexual activity, courtship, and nesting behavior. Pars intermedia is the little sliver in between the anterior and posterior pituitary. Melanocytes are the cells in the skin which help to block nuclei and the nucleus from the sun. They are responsible for the secretion of melanocytes. Block nuclei and nucleus from the sun by absorbing light rays. Constant producing. Rises and falls in response to the sun. Response to influence from gland. MSH, melanocyte stimulating hormone, is under the influence in the bloodstream, causes integumentary system to produce more melanocytes and melanin. Involves light, more sunlight. An example, more sunlight causes more melanin to be produced from the skin and more melanocyte stimulating hormone. Next figure. Next figure, the hypophyseal-portal system and the blood supply to the pituitary gland. So we have some supraportic paraventricular nuclei, and these nuclei lead to arteries, capillaries that lead to the endocrine cells. Okay, ADH and oxytocin are sent through neurotransmitters, hormones going through the adenohypophysis to the bloodstream. The hypothalamus sends ADH and oxytocin through the neurons as if they are neurotransmitters, but hormones going through adenohypophysis needs to be secreted by the hypothalamus into the bloodstream, but must only go into the anterior pituitary adenohypophysis. So we create a portal system. So going through adenohypophysis needs to be secreted by hypothalamus into the bloodstream, only into the anterior pituitary. So we design a tiny portal system. The nutrients go into general circulation of duodenum from the nutrients go to the liver, where it decides what to do with the amino acids. And portal blood goes straight from the hypothalamus. And portal blood, the hypophyseal instead of hepatic portal system. Oh! In portal blood, hypophyseal instead of hepatic portal system. Hypophyseal instead of hepatic portal system. Goes straight from the hypothalamus to the adenohypophysis, where it delivers releasing hormones. From the hypothalamus. Delivery is due to hypophyseal portal blood circulation system. Short circuit to grab oxytocin and ADH. Hypothalamic releasing of hormones gets released, goes into the blood circulation system. ADH, oxytocin, melanocyte stimulating hormone. 6 purple boxes. So these 6 purple boxes in the pituitary hormone and their targets: hypothalamus, their controlled nervous system. Some direct control of the nervous system, indirect control to release of regulatory hormones. Direct release of hormones. So we have adrenocorticosteroid, adrenocorticosteroid hormone. It's from adenohypophysis going to adrenal gland, epinephrine, norepinephrine production. Thyroid stimulating hormone goes to thyroid gland, produces T3 and T4. Also, ACTH produces glucocorticoids such as cortisol and corticosterone. Growth hormone goes to the liver, portal system, or blood circulation system, and then produces somatomines, produces growth of bone, muscles, upper tissue, cartilage, and all that. Prolactin is produced, goes straight to mammary glands. Follicle stimulating hormones and luteinizing hormones go straight to the testes and ovaries. Inhibit and then produce more hormones, inhibin testosterone, estrogen, progesterone, and inhibin. The hormone ADH activates the kidneys for water reabsorption. Oxytocin for males, smooth muscle and ductus deferens. Females, uterine, smooth muscle, and mammary glands. So, indirect control through release of regulatory hormones. Regulatory hormones are released through hypophyseal portal system. Regulatory hormones are produced through the hypophyseal portal system for delivery to the anterior lobe. Of the pituitary gland. Direct release of hormones, sensory stimulation, and osmoreceptor stimulation. So the responses to releasing hormones. Next was AD. Releasing hormones are always have capital R, allow anterior pituitary to respond correctly. Tropic releasing hormone. This releasing hormone ultimately ends up speaking to adrenal gland, speaks to adrenal gland and thyroid hormone. Releasing means coming from hypothalamus and instructing the anterior pituitary to release something specific. Using the chemical hypothalamus and instructing the anterior pituitary to release something specific. Okay, thyroid releasing hormones, HTRH, adrenocorticotropic releasing hormones. So adrenal cortex speaks to adrenal cortex and thyroid gland through the adrenal hypophysis. PR, prolactin releasing factor. Prolactin speaks to the mammary glands, ultimately factor, not hormone. Gonadal releasing hormone, gonadotropin, speaks to gonads, ovaries, and testes. Somatotropin releasing hormone covers the releases. Covers the releasers. Be familiar with releasers in OH. One releases from the adrenal hypophysis. Wow, that's a lot of information. And now I have reached my latest information, the latest day of lecture. We covered a lot of information. I'm going to go through them all today. Let's do this. So the three regions of the hypophysis, the recap of yesterday's quiz. Adrenal hypophysis secretes releasing hormones. Pars intermedius secretes MSH, melanocyte stimulating hormones. Neurohypophysis as supraprincipal functions secrete ADH and oxytocin. The three hormone structured as lipids include aldosterone, testosterone, calcitriol, estrogen, androgens, and cortisol. 3. A releasing hormone releases, it's going to release a tropic hormone stimulating hormone speaking to another gland to release another hormone. Releasing hormones release their own set of hormones that go to other glands rather than going to an effector. Release stimulating hormone that asks other glands. And then a recap on an old quiz again. Nearsight, distant vision means your shape of your eyeball could have a problem because the eyeball can't be too round, short, or long, long, short. For nearsight, it means you have a long eyeball when the lens is round, fine, but can't focus enough for distant vision. Transduction is the translation or converting of a physical stimulus into the language of a neuron or action potential. Stimulus include tread rotation, acceleration, gravitation, gravitational force, olfaction, gustation. Know what form the stimulus arises at a receptor to result in an action potential. Is a hair bending or is a rod using its photopigments to absorb light? Transduction happens at the point of reception caused by bending of hair. Round, long, short. If the lens won't bend around, then you need a long eyeball for light to reach fovea. Close objects require more bending of lens. Good close vision, not good for distant vision. Eyeball too long for distant vision. For farsighted / close vision, opposite lens won't round enough. Lens rounds fine but can't round enough to focus for distant vision. The eyeball is going to be either really long or the lens will be very rounded. The eyeball is either going to be really long or the lens will be very rounded to make you myopic. If your eyes focus better on an object, if your eyes focus better on an object close and not well on an object far away, it means you are nearsighted and have poor distant vision. Caused by the shape of eyeball, could be too round, long, or short. Same figure on the pituitary hormones and their targets. Releasing hormones of the hypothalamus, speaking to the anti-pituitary in the language of releasing hormones. The neurohypophysis posterior pituitary gets a neurosignal from the hypothalamus. Tropic hormones can also be written as trophic hormones. Some hormones have a T for trophic or S for stimulatory. Speaks from adenohypophysis to the bloodstream to another gland. Trophic hormone in liver, adrenal gland, thyroid gland, liver, mammary, testes, ovaries. Category speaks to gonads, mammaries, liver, thyroid, and speaks to portions of the adrenal gland on list with releasing hormone story. Releasing hormones include back to the feedback control of endocrine secretions figure. Feedback releasing hormones include prolactin releasing hormone or prolactin releasing factor, the same thing. From anterior pituitary to the adenohypophysis to the mammary gland. It speaks to modified version of the sweat gland. Prolactin releasing hormone goes into the bloodstream, the mammary gland, and the mammary gland releases prolactin. Prolactin is a hormone which tells mammary gland to release hormone. Mammary releases hormone prolactin. Prolactin tells the mammary gland to release its own hormone. Prolactin tells the mammary gland to release its own hormone. Gland sweat, milk secretion, modification of sweat from the sweat gland. Ultimate effect is to stimulate milk production. Doesn't project milk until oxytocin does its role. Doesn't project until smooth muscle contract in the mammary gland. Prolactin is only good if the oxytocin is making the ejection happen. Prolactin inhibitory hormone can turn on or off tropic hormone secretion. When inhibitory hormone is secreted, GHIH, gonadal hormone, inhibitory hormone, as that increases secretion. Mammary glands have milk-producing glandular tissue and also have smooth muscle so that milk can be projected out of the glands. Growth hormone speaks to the liver, the hypothalamus to the anterior pituitary to the gland. So the gland example would be the liver receiving growth hormone, and then somatomedin, STH. Somatomedin, tropic hormone, tropic coming from adenohypophysis, speaking to another gland. Tropic coming from adenohypophysis, the gland here would be apparently the liver. Example is be from the liver, secretes somatomedin to an effector, which could be multiple. Effector is muscle cells, cartilage grows, adipose tissue, muscles grow at a cellular level. Chondrocyte cells and cartilage, adipose cell to multiply. Protein synthesis is lipid and glycogen breakdown. Cell division, gonadal hormone, inhibitory hormone stops or slows the production of growth hormones. Reduce frequency in growth processes. Reduces the frequency in growth processes. Supply roots. The supply routes are inhibited when inhibiting growth. Inhibiting growth, reducing insulin secretion. Don't let glucose in so the cell can't grow. Not letting glucose in so the cell can't grow. Insulin allows glucose in to work with the mitochondria needed for a cell to do what is needed. Okay. Take a quick break. I'll be right back. Holy cow! Yeah, my screen's playing some loud music. It's not the good kind. Okay, let's finish this. Been at this since 5 a.m. I'm about a couple more minutes left until my exam. So, next figure is on the thyroid gland. So you can see the thyroid gland here, right there. No thyroid vein, follicles, thyroid follicles, capillary. A bit of a cross section of it. And a microscope view. Thyroid releasing hormone comes from the hypothalamus. It speaks to the adenohypophysis in response to the thyroid release. Thyroid stimulatory hormone TSH specific to the thyroid gland. Stimulating tropic speaks to another gland for that gland to produce an effect. In the thyroid gland, this that consists of colloid substances, contains thyroid hormones. Thyroid cells are secreting hormones in the world of thyroxine. Number T3, T4, thyroid hormones that go out to body cells to increase cellular metabolism and growth rate. higher than normal blood pressure. Higher than normal blood pressure increases ATP, increases red blood cell production, body temperature. Thyroid hormone does everything cells need to do to step up metabolic pace to increase overall metabolism. Growth hormone cells to help body grow. Okay, next figure is going to be for the adrenal gland. Okay, so the adrenal gland layers as a capsule, an outer cortex, wide middle layer, inner layer, blending in with the medulla. 4 parts of the adrenal gland are cortical layers. 2 parts of the adrenal gland respond to releasing hormones from the hypothalamus, middle, and inner. Adrenal cortical releasing hormone, ACRH, speaks to adenohypophysis releasing adrenal cortical thyroid hormone, going into the middle layer of the cortex of the adrenal gland. Adrenal gland mid-cortex is responding light. The response for releasing of hormones, cortical and cortisone. Responsible for releasing of hormones, cortical and cortisone, glucocorticoids. Involved in processes of the thyroid gland. So it's involved in the processes of the thyroid gland by increasing the synthesis of glucose coming from glycogen, lipids, and proteins from places such as storage or diet, increasing immediate supply of energy sources for all cells that are about to use them. Increasing synthesis of glucose coming from glycogen, lipids, and proteins from places such as storage or diet, increasing immediate supply of energy sources for all cells that are about to use them. Supply chains. Our body cells can't do a lot with proteins. Get storage ready. Cortisones and cortisols reduce inflammation, presented hand in hand with stress response. Because epinephrine does the opposite when presented with a stress response. Epinephrine increases immunity. Make sure supplies are there and that you are not taken over the edge. Breaking while assisting stress response. Glucocorticoids are involved with stress hormones secreted under stress. Exists in a world of stress. Deal with situations when we are under stress and can't resolve it. Negative effects of glucocorticoids. Place to be supportive effects. Inner cortex includes androgens. Androgens are also part of the releasing hormone story. Adrenocorticotropic releasing hormones, ACTRH, released from the hypothalamus, speaks to the inner cortex. Adrenocorticotropic releasing hormones released from the hypothalamus speaks to inner cortex. Release androgen family, mainly testosterone. Release androgen family, mainly testosterone. Goes to target organ, bone, muscle, hypothalamus. Increased bone density, muscle, sex drive, testosterone issue, hypothalamus, testosterone, estrogen. Glucocorticoids on the outer cortex responsible for secretion, ultimately aldosterone, not part of releasing hormone story. Aldosterone, not part of story. Okay. Next figure. Alright, the general adaptation syndrome. So the 3 phases. So we have an alarm phase. During the alarm phase, an immediate response stress occurs. Sympathetic division of autonomic nervous system directs this response. Energy reserves mobilized in the form of glucose. Body prepares to deal with stress-causing factor by fight or flight response. Epinephrine is the dominant hormone alarm phase. Its secretion is part of generalized sympathetic activation. Brain sympathetic activation, epinephrine. Immediate short-term responses to crisis increases mental alertness, increases energy use by all cells, mobilization of glycogen, lipid reserves. Changes in circulation, decreased digestive activity, increased sweat gland secretion, increased heart rate, and respiratory rate. Resistance phase, if a stress lasts longer than a few hours, the person enters the resistance phase of the general adaptation syndrome. Glucocorticoids are the dominant hormones of the resistance phase. Epinephrine, growth hormone, thyroid hormone. Energy demands in the resistance phase remain higher than normal. Due to combined effects of these hormones, neural tissue has high demands of energy and requires reliable sources of glucose. If blood glucose levels fall too low, neural function deteriorates. Glycogen reserves can meet neural demands by the alarm phase, but becomes depleted after several hours. Hormones of the resistance phase mobilize lipids and amino acids as energy sources to conserve glucose for use by neural tissue. Sympathetic division, growth hormones, pancreas for glucagon, ACTH, cortex for glucocorticoid, mineral corticoids, ADH, renin, angiotensin, aldosterone system from the kidneys. Long-term metabolic adjustments, mobilization of remaining energy reserves, lipids are released by adipose tissue, amino acids are released by skeletal muscles, conservation of glucose, peripheral tissue, except neural. Break down lipids to obtain energy elevation. Blood glucose concentration. The liver synthesizes glucose from carbohydrates, amino acids, and lipids. Concentration of salts and waters, and also potassium and nitrogen. Exhaustion phase. The body's lipid reserves are sufficient to maintain the resistance for weeks or even months, but when the resistance phase ends, homeostatic regulation breaks down and the exhaustion phase begins. Unless corrective actions are taken almost immediately, the failure of one or more organ systems proves fatal. The production of aldosterone throughout the release. The resistance phase results in production and the conservation of sodium. The production of aldosterone throughout the resistance phase results in the conservation of sodium at the expense of potassium. As the body's potassium contents decrease, a variety of cells begin to malfunction. The underlying problem of the exhaustion phase is the body's inability to sustain the endocrine and metabolic adjustments of the resistance phase. Collapse of the vital system, exhaustion of lipid reserves, cumulative structure or functional damage to vital organs, and the produce glucose, failure of electrolyte balance. 3 phases of stress response. We are built to respond to emergencies with an immediate response where stress is first recognized, and sympathetic division of the autonomic nervous system directs energy reserves to mobilize the body to prepare itself for fight or flight. First phase is alarm phase, increasing mental awareness, change in circulation, peripheral vasoconstriction, and vasodilation, increased energy use by all cells, energy put in form ready, mobilization of glycogen and lipid reserves, increased sweat gland secretion innervated by sympathetic division, not parasympathetic division, increased heart rate and respiratory rate, dilating tiny capillaries and around lungs. Get oxygen and glucose to the heart to be pumped to the brain and lungs, also to the muscles. Decrease digestive activity and urine production. Resistance phase, less epinephrine effect, more glucocorticoid effect, making sure body can maintain its self-conservation of salts and more loss of potassium and hydrogen. Ensure you don't overdo it. Make sure you have the supplies. You can't do first phase without the second phase. Constant access to food, constant eating, exhaustion phase. You can't keep doing both. In the previous 2 phases about exhaustion, you're exhausting yourself. Lipid reserves are gone. You used up lipids and protein reserves. Electrolyte balance is out of whack. No reserves, eat body proteins. Electrolyte balance go crazy. Sodium and potassium exchange results in decreased potassium cell malfunction. Balance crazy if you cannot replace it. Last figures, the pancreas. In addition to being a digestive exocrine organ, it is also an endocrine organ, secreting insulin and glucagon. They get involved in metabolism, storing, and respiratory distress. Glucose sensed in the blood responds in the pancreas, stimulus increase or decrease blood glucose level. 2 flow charts. Beta cells secrete insulin if glucose levels in blood are high. Stimulus increase or decrease. Alpha cells secrete glucagon if blood glucose levels in blood is low. Insulin speaks to all. Nervous system can only use glucose, while some cells use insulin to take glucose in. Negative feedback situations, too much insulin gets so much glucose into cells and blood gets taken back to normal levels. Negative feedback systems get you back to normal levels. Results of today's glucose and insulin review about type 1 and type 2 diabetes. So types of diabetes, okay. Type 1 diabetes is characterized by inadequate insulin production by the pancreatic beta cells. A person with type 1 diabetes requires insulin to live and usually requires multiple injections daily or continuous infusion through an insulin pump or other devices. The form of diabetes accounts for approximately 5% of cases. It usually develops in children and young adults. Type 2 diabetes, so they're not getting enough glucose and they need more insulin. Type 2 diabetes is most common for performance diabetes mellitus. Most people with this form of diabetes produce normal amounts of insulin, at least initially. When the tissue does not respond properly, a condition known as insulin resistance. Type 2 diabetes is associated with obesity, and weight loss through diet and exercise can be an effective treatment. Okay, results of today's glucose issues. We have about type 2 diabetes, that type 1 insulin not produced at right levels, genetic components produced by lifestyle habit, not exercising, 2 types of diabetes. Diabetes type 2 inflicted upon yourself by lifestyle of habit involves injection of insulin that requires treatment and change of lifestyle. 18-12, parathyroid hormone, calcium regulation story, calcitriol and calcitonin. Parathyroid glands are evolved in the back of the thyroid, secreting parathyroid hormone, secreting calcitonin and calcitriol. Calcitonin is secreted by the thyroid gland and works to maintain calcium levels in the blood. Parathyroid and thyroid work to maintain calcium levels. Parathyroid hormone and calcitriol from the kidney make sure blood has enough calcium Make sure blood has enough calcium reabsorbed from the kidney and out intestines. Okay, that's all for today. I have to take my exam in a few minutes, so let's hope for the best. Thanks for watching. God bless.